Hey, thanks for checking out SGU TV. Today we're going to talk about diapedesis, or otherwise known as leukocyte extravasation. Now, what is this all about? It's about white blood cells, which normally travel inside of our bloodstream, and how they're able to actually leave the bloodstream. Our heaviest particles will travel in the middle of the stream. Looking at the blood, who's going to be the heaviest? Well, it's going to be actually the white blood cells that are hanging out in the middle. And then on the outside will be red blood cells. Then after that, we're going to have actually some platelets. Then we're going to have the plasma traveling in the middle. We'll be rolling along the surface over here, right? And that's because of due to something called over here. You see those little purple cup-shaped things. Okay, over here it's larger, right? And we can see that these things are actually called P-selectins. Okay. On this side of the chart over here are all the different types of devices that will be hanging on the actual lymphocyte itself. Okay, and over here, all these items over here will be located on the actual endothelial surface. Okay, so you can see that the P-selectins over here, these purple-shaped things, are attaching to these oligosaccharides. Like I said before, about half of these white blood cells are actually rolling along the surface, about half of them traveling up here. If we have an infection over here or some type of inflammatory response, we will have the release of IL-1, TNF-alpha, and C5a. IL-1 standing for interleukin, TNF standing for tumor necrosis factor. Okay. Remember, if we have tumor necrosis factor, we expect to be releasing inflammatory markers. C5a, that's from the complement system. This will also be encouraging inflammation. And these signals coming out from this particular infection, right, will actually cause more P-selectins to come to the surface. And therefore, a larger percentage of these lymphocytes will actually roll across the surface here. Now, this lymphocyte over here, I have it actually looking like a pinwheel because it's rolling, right? And you can see that I have the blue sugars on the end, and I have the purple cups that's attaching to the attachment is happening, right? And like I said, P-selectins are attachments to the oligosaccharides. You can see generally these molecules will attach to these molecules, right? Actually, these ones attach to these ones because this is going to be on what? On the lymphocyte over here, and this is going to be on the surface here, right? So the P-selectins are down here, connecting the oligosaccharides. Next, we have something called PAF, okay? PAF is also known as platelet adhesion factor, okay? And we actually see this done in green over here, and we can see it's hitting the PAF with a platelet activation factor receptor, right? So PAF in green and the PAF receptor. Okay, so we know that they're going to be rolling along, but sometimes, right, this will have it lined such that the PAF will activate this receptor, which will actually activate these guys over here, right? And these guys are known as integrins. Okay. So these integrins, okay, are made up of actually LFA1 or MAC1, right? Now these guys over here, these specifically attach to the ICAM1, right? And here are a pair of ICAM1s, right? The VLA, which stands for very late antigen, actually attaches to VCAM, or the vascular adhesion molecule. Let's talk more about ICAM1 and these particular guys that it's attaching to, and exactly what its role is in this whole entire procedure, right? Over here, we talk about the very first thing that's happening. We talk about the P-selectins attaching to the oligosaccharides. Then we said that the platelet adhesion factor would hit the PAFR, the platelet adhesion factor receptor here. This is going to stimulate these integrins over here, these red guys. Okay, talking about LFA1 or MAC1. And one thing we have to remember is that these guys are CD11A and CD18, right? Cluster differentiations. So you can see over here, there's a pair of guys over here with their hands like this, right? They're looking kind of tough, and we can see that the word dad, okay, and tough, right? Tough dad A and tough dad B, right? Tough dad A is laughing. You see this laugh coming out of his mouth, right? And he's not going ha ha ha, he's going to laugh, 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 because we're not going ha ha, we're going to laugh, right? Or LFA, right? We're going to slick it around. The second daddy over here has a fancy little piece of hair piece coming off his head right here. So he's a Mac, right? So he's daddy type B, right? Okay. And if you remember why I'm telling this one daddy, right? I'm using the major system to translate these numbers into letters, and then into sounds that I can memorize these particular concepts off of, right? We can see over here that these two, right, these were the eye cams, right? These two big eye-looking shaped things, right? And they're connected underneath by a V-shaped mechanism, right, kind of holding them together, so they'll be correct for this thing to fall into place. This V stands for the V-cam, or the vascular adhesion molecule. Okay, remember that this is actually located in the vasculature, right? Okay, this is also located in the vasculature, but remember this is the VLA4. Okay, and this one is the last one to add itself to the party over here, right? These whole things assemble. This thing activates this thing. It comes down. This is the last piece to activate and attaches to these guys over here, which has the I and the V, so the I cam and the V cam, right? So after this happens, right, these molecules over here, these ones, remember, are just transitory bonds, okay? These are just to roll across, right? These green ones and the blue ones are activating, right? I don't really have that marked up here, but these red ones and blue ones here are actual true adhesion. They're going to get stuck there. You see these two over here? This leukocyte, which kind of like has planted itself here with these two legs that are stretching down. Those two legs are actually made of, right, these CD14 and CD11 and 18 combinations, either LFA or MAC, right? And the ICAMs, right? And then from here, we can see that the cell is poking and piercing its way through, piercing and pushing its way through the wall here. And why do I do that? Say poking, piercing, and pushing through the cell wall? By remembering those three P's, right, it kind of gets me back onto the whole thing. So I think about a blood vessel, I need to remember the fact that this is rolling down, right? And then I say, oh, okay, what's happening with this white blood cell? It's going to be poking, piercing, and pushing its way through the cell wall. Then I remember, oh, we started again with the P-selectins and then the PAF, right? So this cycle just brings me back to the whole thing of where I'm starting. It helps me remember how to put everything into order for this particular piece. I hope that this would help for you to help memorize all the different pieces of this little structure here. I know it's a little tricky, especially when memorizing these little guys, okay? But it was helpful for me. I think it'll be helpful for you. Thanks for checking this out. Alright, that's it. Thanks for checking out SGU TV. Please make sure to like, comment, and subscribe, and check out our other videos.